Well, hello everyone, and thank you for joining us for today's webinar entitled Tissue Microenvironment, Biochemical and Biomechanical Changes and Impact in Chronic Lung Diseases. My name is Richard Johnston, and I will be co-moderating today's webinar along with Sanjana Mahadev Bhat from the Mayo Clinic. Today's webinar will highlight the innovative research of Dr. Janette Burgess, recipient of the 2025 Solbert Permutt Trailblazer Award. This award recognizes, or it's bestowed by the ATS Assembly on Respiratory Structure and Function, and it recognizes an established investigator who has contributed pioneering research in respiratory structure and function and has guided and inspired others in the field. Briefly, by way of background, Dr. Solbert Permutt was an intellectual giant who made groundbreaking contributions to our understanding of pulmonary physiology and respiratory medicine. He was also an extraordinary mentor who inspired generations of trainees and colleagues. So I'd now like to make way and introduce our featured speaker today, Dr. Janette Burgess, who is currently Professor of Extracellular Matrix in Disease Pathogenesis and a Rosalind Franklin Fellow at the University Medical Center Groningen in the Netherlands. Dr. Burgess received her Bachelor of Science with honors in biochemistry and microbiology and immunology from the University of Adelaide in Australia. She subsequently earned her PhD in molecular biology from the University of New South Wales, also in Australia. Although her doctoral work focused on thrombosis and vascular disease, her entry into respiratory biology began with a postdoctoral fellowship at the University of Sydney, where her research focused on gene expression in asthmatic airway smooth muscle cells. So following her postdoctoral fellowship, Dr. Burgess has remained at the University of Sydney, where she rose through the ranks and ultimately became an associate professor. In 2015, she departed Australia for the Netherlands to assume her current position. Now, for more than 2 decades, Dr. Burgess's research has been continuously funded by a range of government and private foundations, reflecting the rigor and innovation of her work in respiratory biology, specifically investigating the extracellular matrix in both the development and the progression of chronic lung diseases. Dr. Burgess amazingly has authored more than 240 scientific manuscripts, reviews, and book chapters, and in 2007, she was the recipient of the Anne Woolcock Memorial Award from the ATS Assembly on Respiratory Structure and Function. Dr. Burgess additionally has also been highly engaged in the ATS and was the previous chair of the Assembly on Respiratory Structure and Function. In addition, she has chaired scientific sessions or served as a speaker at numerous scientific conferences, more than 90 conferences or academic medical centers. Now, part of the award, the Trailblazer Award, is that the recipient inspire and train mentees. And Dr. Burgess is deeply committed to training the next generation of respiratory biologists. She has served, again, incredibly, on more than 60 student thesis committees and mentored the research activities of more than 80 undergraduate, master's, or PhD students, and has mentored 9 postdoctoral fellows, all while maintaining a substantial teaching portfolio. Now, although I could continue going on highlighting Dr. Burgess's many outstanding accomplishments, I will stop here and turn the program over to her And I want to say I've personally had the pleasure of working with Dr. Burgess over the past 15 years on several ATS activities, and it is truly an honor to introduce her today. So, Dr. Burgess, congratulations on the award, and the floor is now yours.
Thanks, Rich. I'm a little humbled by that introduction. So I think we'll just move straight into some fantastic science. So it's my pleasure today to talk about tissue microenvironment, as Rich indicated, and how this has an impact in chronic lung diseases. These are my disclosures that are relevant to the presentation. So we're all interested in lung diseases and how we can find ways to alleviate these. And when I have conversations with people, sometimes they are inspired to make drawings. So this is one take on what a diseased lung looks like. And I'm particularly inspired by the obstruction that's illustrated here. This is a second take, which was actually painted by an artist who also happened to have pulmonary fibrosis. And they explained that the dark spots they illustrated were where they felt they couldn't breathe, whereas there were regions in their lungs that they could breathe through, which kind of illustrates the heterogeneity of the changes that we're dealing with in these lung diseases. So my team are interested in the structure of the lung that supports all the cells that we talk about. What we're looking at here are images taken with a special microscopy technique called second harmonics generation that highlights for us collagen fibers. So the pink and the blue on the left and the orange and the green on the right, and I'm sorry, the colors are different. The students got carried away with their color coding, are showing us collagen fibers with different structures. And the yellow or on the left is showing us airway smooth muscle because this is the lumen from an airway. And here we're looking at the alveolar region. So you can see that these fibers are very arranged and they're spatially organized. So there's a lot of structure in this. And when I talk to students about what is this extracellular matrix stuff that surrounds cells, these are the words that they come up with. They think about collagen. Sometimes we think about fibronectin, elastin, but it's a support structure. It forms a scaffold. It binds growth factors. It has mechanical properties, and all of this influences how cells respond to it. And the reason that I'm intrigued by this is that it changes in these diseases that we think about. When we compare a non-diseased airway to an asthmatic airway, the picture looks different. So we're looking at, in the green, collagen, or in the lower pictures, the blue and red is showing us collagen. And you can see, particularly in this IPF tissue section, that where there's nice open spaces in the non-diseased lung tissue, this is filled with this extracellular matrix in IPF. And COPD is an even more intriguing picture because we've got areas around the airways that are thickened, so matrix deposited. But we know that in emphysema, we've got destruction of the alveoli, so destruction of this support structure. And my team want to understand how these changes can then influence disease processes, so influence these cells.